Hello everyone, hi. Olá a todos, sejam todos bem-vindos a mais uma sessão dos nossos webinários internacionais. E hoje nós temos o prazer de contar com um convidado dos Estados Unidos e por isso o idioma deste webinário será em inglês e toda a comunicação. So, hi everyone and thanks for being interested in watching this amazing lecture and that's about to come. And we have today here Professor Stephen Maxey from the School of Biological Science at the University of Northern Colorado. He's going to talk about the evolution and diversification of snake venom toxins and venom glands. And it's a great pleasure to have you here, Professor Stephen. And on behalf of the Graduate Studies and Research Office of Unifesp, we would like to thank you. But we also would like to thank you, Professor Miriam. Professor Miriam is a tremendous partner of the Graduate Studies and Research Office. She's been dedicating her time to organize a lot of international webinars. And for sure today, it's going to be another one with a very successful interaction between our inviting and our public. And Professor Dr. Miriam Hayashi, she was born and educated in Brazil, and she's currently Associated Professor of Pharmacology at Unifesp and head of the Molecular Pharmacology Lab at the Department of Pharmacology at the Escola Paulista de Medicina. Professor Hayashi is also vice-chief of the discipline in cellular pharmacology and member of the scientific board of the Intellectual Property Office of Unifesp. She completed her pharmacy and biochemistry degree at the State University of Londrina in Paraná, and she received her PhD at Unifesp and postdoctoral fellowship in molecular biology at Butantan Institute, which is a Brazilian institution mostly renowned by the scientific research with venomous animals and production of vaccines. And Dr. Hayashi has published more than 90 scientific articles on mental disorders and natural compounds for drug discovery field, besides several chapters of books and patent applications filed. And she's also she was also recently awarded with the prize from Fleury Group Innovation Award, and thanks to development of a method of identifying biomarkers for severe mental illness by nuclear magnetic resonance and chemometrics. And so she is a very important, and today we have a very important topic to discuss here because it's a topic in between the most prestigious area of Unifesp, which is the health area, but also we have a lot of biodiversity researchers, and I'm pretty sure our students, our post-graduation students and grad students, they are here for listening to this amazing. Thank you so much, both of you, Professor Miriam and Professor Stevens.
You're welcome.
Thank you, Camilo, for this great introduction. Now I go with Dr. Stephen. So first of all, thank you very much for accepting this invitation. Dr. Stephen Maxey is a professor of biology in School of Biological Science at the University of Northern Colorado. His research broadly encompasses the biology of venom snake and the biochemistry of snake venoms. And he has published more than 200 scientific papers, book chapters, and natural history notes, including several books. One of them is the Reason That We Know Each Other. That's named the Handbook of Book and Toxin of Reptiles, CRC Press. So if you are interested, please buy this book. He's going to release a new edition this year. And his research has included many graduate and undergraduate students, as well as a collaboration with colleagues from Singapore, Spain, Mexico, Costa Rica, Argentina, Brazil, France, India, and others, including various other universities in the USA and University of Texas. Several ongoing projects are centered on understanding the evolution of venom system in snakes and the biological significance of venom composition and variation, particularly in areas of introgression and hybridization, with a special interest in interface between snake ecology, evolution, and venom biochemistry and pharmacology. So, Dr. Stephen, sorry for my awful English, and I hope
Your English is fine.
I could just highlight the main achievements. Your CV is really great and long, so we could stay like half an hour reading it in my bad English. So I will give the screen for you, and thank you again for your invitation.
Well, it's very much my pleasure to be here. I wish I was there in person rather than electronically, but this is our life these days. So I'd like to thank in particular Professor Hayashi for the invitation to talk to you today about some of the work that we do in my lab. And some of the things I'm going to talk about today started a long time ago and have extended into recent times as well. So hopefully you'll find some interesting things as we go through talking about evolution and diversification of venoms and venom glands. And excuse me if I constantly am clearing my throat.
Yeah,
but
Stephen, just sorry for interrupting you. I forgot just giving an advertise for the attendees. So you can make questions in the chat. So you write the question and you can read the questions for Dr. Stephen that is going to answer at the end of this presentation. Okay, thank you. I'm sorry. Go ahead.
No problem. Yeah, please do ask anything, and I'll give my best attempt to try to answer it. Before we get started, though, I thought I'd show you a little bit about what things are like right here, because Colorado is a bit different from Sao Paulo at the moment. And let's see if so this is what we're looking at today in Colorado, and it's supposed to be spring, but yesterday a snowstorm came in and coated everything with snow. So even though we're going into spring, we still have vestiges of winter, and it was quite cold this morning. So anyhow, I get this question a lot: why study venoms? And to me, it's kind of a strange question, a non sequitur. Of course you should study venoms, they're very interesting. But if we think about it, venoms are these very potent biological products that have toxic effects. And so that, of course, just in itself is rather interesting. If you're more evolution and ecologically minded, these are characteristics that have direct fitness components as well as implications. And so if you think about that, the snakes are producing these toxins, they're immobilizing their prey with it, and so they're obtaining food by using these remote delivered chemical means of predation. We can also look at this from a chemical predatory sort of strategy and ask questions about why venoms vary from this snake to that snake. Why are coral snakes and rattlesnakes so very different in their venom composition? And along that line, that provides an open area for all sorts of potentials for drug discovery and interesting new pharmaceutical compounds that may in fact have therapeutic applications. So if you're more interested in esoteric biochemistry, protein evolution, and aspects of how toxins in fact have evolved over time, developed new activities, neofunctionalization of these molecules is also an area. And one thing I'll talk a little bit about today is structure function type of studies. So how we can have these molecules that on the surface look very similar, but small changes lead to very large changes in their biological activities. So what I'd like to do is start out giving you a rather extended overview of venomous snakes, talk a little bit about the diversity of species that we see worldwide, a little bit about the venom apparatus that is used to produce and deliver these compounds, and then talk a bit about what we can call venom compositional strategies. Okay. To do this, I'll use two vignettes from work that we have been conducting over some time, really, and some of the earliest of this started 20 years ago and has extended until current times. So we'll really look at a kind of a combination of features that provide an integrative biology approach to things, and then talk about three-fingered toxins, a particular family of compounds with some very interesting properties. Okay. Finally, then I'll try to wrap this up, summarize, and leave you with some take-home conclusions. Okay. So first, let's take a look at venomous snakes and their venom apparatus. And so just to give you a feel for the diversity of the actors in this whole scene of what we call toxinology, the study of venoms, there are a wide variety of different types. So we've got old world vipers that range in size from very large to very small bodied. We have old world pit vipers that are characterized by heat sensitive pit organs, very diverse group of these all through Asia. We have in the New World, sometimes in your area, your general vicinity in that part of Brazil, a wide variety of pit vipers that are highly specialized as arboreal snakes or terrestrial snakes. In the New World, we have also a wide diversity of rattlesnakes, and these reach their apex in Mexico, but they extend all the way from Canada into central South America. So very broad range and very interesting diversity of species. And a lot of work that we've done has included these various species of rattlesnakes. And again, some of them highly specialized. All these are mountain forms. And then there are lowland tropical rattlesnakes that have much larger body form. Okay. This contrasts rather distinctly with the elapid snakes, things like cobras, the coral snakes, and their allies, and very, very different venoms. We'll talk about those briefly, but we're going to spend a bit of time talking about rear fang snakes in several different families, including the Colubridae. And they also are very, very diverse in their form and their distribution globally, their habits, and their basic biology. And so another family is the Dipsatidae, and I'm not going to belabor the taxonomy because higher snake taxonomy has been a point of contention for many, many years for many people. Suffice it to say, there's quite a diversity of forms. And so what we see this is a cladogram that was published several years ago by other people. And in fact, what we have is a wide diversity of different types of species globally. And if we ask the question, where do we find venoms? Well, the vipers, as we've just seen, are a reasonably diverse group of animals that range all the way from the tropics to the subtropics and temperate regions. The elapids, more of a tropical, subtropical group. And there's a fair diversity of species here, 300 or 400 species total. When we look at the so-called colubrids or the rear fang snakes, there is a tremendous diversity of species, and the majority of the advanced snakes fall into these two families groups, or three family groups, depending on how you want to parse them. So we have a lot of different species potentially to work with, and in particular, this group is much less worked on than the vipers and the elapids. Okay. So if you're interested in any aspect of biology, there is a particular subdiscipline of biology that is involved in the study of venoms. And so venoms have become a very popular area for research, regardless of whether you're more morphology-based, comparative anatomy type of things where you're looking at the structure of the glands themselves. Again, if you're more evolution ecology-based, or if you're really focused in on protein chemistry, the venoms are primarily proteins, so lots to do in that area. Proteomics, transcriptomics, and genomics have, of course, blossomed and taken the front stage in many areas of biology. And of course, these are very, very important disciplines for understanding venoms, venom evolution, and venom structure-function relationships. So regardless of where your interests in biology are, there's probably some niche there that includes things like the study of venoms. Okay. One of the things that I was first interested in as a graduate student and then still continue to this day is looking at the structures themselves. Front fang snakes, such as rattlesnakes in the genus Crotalus, have a very large reservoir of venom in this venom gland. It is delivered with a very high pressure at a very rapid rate. And so the whole strike sequence, strike envenomation and retract, takes somewhere on the order of 250 milliseconds. Very, very fast, delivered through these teeth that are hollow and are very much like a hypodermic syringe needle. If we contrast that with the rear fang snakes, we have a much lower pressure system, and this is delivered via a gland that has similarities to the venom gland of front fang snakes, but typically lacks any kind of muscular push to deliver the venom at a rapid rate. And so it's a relatively slow, low pressure delivery system that's associated with enlarged teeth on the back of the maxillary bone here. And here we can see that in larger detail. It's still very distinctive histologically from the salivary glands that are mucosecretory, and these are serous secretory, producing the venom proteins. Okay. That also affects how we get the venom from the snakes. Front fang snakes, relatively easy. Grab the snake, express the venom into a container. Rear fang snakes, even something like this big boom slang here, much, much slower and much more laborious process, requires a 2-drug regimen to get them to secrete significant amounts of material. And so the venom extraction process is different as well. Well, if we think about what's in the venom, as I mentioned, it's primarily proteins. These are not de novo toxins, but instead they're co-opted compounds that are based on regulatory compounds that are found commonly in the body of vertebrate animals. And they're overexpressed in specialized glands, and in conjunction with Todd Casto's group in University of Texas, Arlington, we've been very interested in looking at some of the molecular mechanisms of how this is controlled and regulated and upregulated very rapidly. So we have a material that is really, really potent as far as its biological activity, but it's really only contains a handful of representatives of protein families, and of the thousands of different proteins that are out there, there's only about 32, maybe 35 that are commonly seen in venoms, and of that, a much smaller number that are the most abundant components. But snakes have done a lot with those venom components, and in a particular molecular scaffold may be highly conserved, but little tweaks and mutations on the surface features have in fact produced very, very different pharmacologies using the same basic molecular scaffold. And we'll look at an example of that in a little bit here. And so by having these non-structural features of the molecule that are basically open to evolution and variation, we can produce things that have very different biological activities, but structurally are quite similar, okay? And it's been recognized a long time ago now that we have greater variation in the portion of the genes that actually codes for the end molecule rather than in introns. And that's kind of opposite from what we see in most standard proteins. So another interesting group of compounds. Now, this is the one you'll want to write down because there'll be a quiz at the end. You'll have to memorize all of these, but not really. What we see is there are a number of different types of enzymatic toxins and non-enzymatic toxins that are found in venoms, but some of them actually predominate. And we'll talk a little bit more about some of those in a minute here. I like to take venoms and do a fingerprinting with 1D-SDS page for a couple of reasons. One, it's very easy. Undergraduates can do this with very little training. But two, it produces a nice picture of the overall complexity that we may be encountering in a venom that we haven't looked at before. And so what we have here is a picture of a gel, mass ranging from high mass here to low mass here. And then on the top here, and in each one of these lanes are different species venoms from rattlesnakes of a variety of different types. And just a quick glance here, we can see that this one contains quite a bit of a P1 metalloproteinase, whereas these species in here seem to lack that one. And so we can get a fair amount of information because we already know quite a bit about where molecules are going to band out on something like a gel. Well, as we've moved into the proteomics era, we can use a wide variety of different types of tools, mass spectrometry, MALDI-TOF and ESI, and a whole host of other different types of technologies have been proliferating very rapidly over the last 10 or 15 years. And these allow us to work with extremely minute amounts of venom. That has led to this idea of venomics, or the study of the venom proteome. And we can start with a variety of different types of materials, but with very, very small amounts, and subject this to a number of different experimental pathways and protocols, and get different types of detailed information from microgram quantities or less of the venom. And this allows us then potentially to catalog the entire proteome. There are some drawbacks to some of these techniques and limitations, but first approximation, this is extremely rapid way to go through venoms and get a good feel for what's in the venom at an individual level, at population levels. And so we can have a deep, deep understanding of venom composition and compare that between species quite readily using almost no material at all. Juan Calvete in Valencia, Spain is very important as kind of a central individual that has really developed and pushed venomics as an area of venom study. And so in a paper that he wrote about 14 years ago here, he lays out the basic pathway, and this is still being used by many people globally to understand venom complexity. Well, as we've gotten into proteomics and transcriptomics and genomics in more and more detail, it turns out that a variety of different approaches will give us the most complete picture of whatever it is we're looking at, and that includes venom composition. And so using next-gen sequencing and transcriptomics and genomics approach and a whole host of other methods, then we can really get at looking at everything that's possible that can be expressed in the venom gland. And so this also favors a high throughput approach where we can deal with lots of samples in a very short period of time. It has the added advantage that instead of having fragments of sequence, we can in fact get complete sequences of the DNA from the genes themselves, but also then translate that into the amino acid sequences. And again, we can address the same kind of questions at individual population levels and broader look at phylogenetic patterns of venom evolution, venom composition. And so by using these various techniques that again have become commonplace now in most fields of biology, we can really probe these systems in much more detail than was ever possible before. Okay. What this allows us to do then is start looking at things and asking the question, well, do we see some patterns? And I'm going to show you some patterns here that have fallen out, seemed apparent to me. This is not absolute. In biology, nothing is absolute. If there are more than one way to do things efficiently, biology finds a way. In the viperids, though, that we've worked with extensively, what we see is what we call type 1 versus type 2 venoms. And there's some very prominent differences between these types of venoms. One, the type 1s tend to be more enzymatically based and result in tissue degradation and all sorts of problems upon human envenomations, whereas type 2 venoms typically lack the metalloproteinase complement, or it's very, very low, but it's dominated by presynaptic neurotoxins. And if we then ask, do we see this kind of pattern in the other groups of venomous snakes? To an extent, we do see the same kind of dichotomies falling out. And again, this is a broad brush generalization, but the elapids are mostly a type 2 venom that is a highly toxic venom that is very dependent on specific toxins, three-finger toxins in particular, phospholipase toxins as well, and very little in the way of metalloproteinases. And we've asked the question about the colubrid snakes. And so what we see is a similar sort of dichotomy in various species. So ranging from metalloproteinases that are highly abundant in the venoms and essentially lacking anything that is neurotoxic to having metalloproteinases as only minor or perhaps not even present at all in the venom, and instead a venom that is dominated by three-finger toxins. And there's some interesting aspects about these that we'll get to in a moment. So just to give you an idea of the picture of this difference in the type 1, type 2, this is just a picture of a composition of various species venoms. And again, a broad brush comparison here. So metalloproteinases, serine proteinases, phospholipases dominate this particular venom. And we see comparable patterns all the way across the board in these species that are pictured here. And these are what we call the type 1, dominated by enzymatic toxins and the SVMPs in particular. contrasting rather strongly with the type 2 that are dominated by things like these beta neurotoxins that are presynaptic in their action. And in fact, some of these, such as the rattlesnake that you have down in your area commonly, is in fact dominated by these neurotoxic components. If we compare those patterns to elapids, we see a very different sort of picture here, and that is the type 2s are found across the board, but we see some interesting differences in the proportions of three-finger toxins and phospholipases in particular, but those two protein families dominate the proteomes of all of these different species, whether we're talking New World species, Old World species, or these specialized snakes, the sea snakes, okay, which have a very, very simple stripped-down venom. Okay. Taking that approach to the colubrids, we have fewer species to choose from, but we see the same kind of dichotomy here, a type 1 enzyme-rich sort of venom versus one that is very rich in specific types of toxins, specifically three-finger toxins. Okay. So these are targeting particular systems in prey animals and in ourselves when we happen to be the recipient of venoms, unfortunately. One of these is the peripheral nervous system. You think about stopping prey, hitting the way that the neuromuscular system is coordinated, it's probably a good idea. And so a lot of different toxins have evolved among these snakes and allow them to knock things out either at a presynaptic site here where we're interfering with acetylcholine release, or commonly at a postsynaptic site here where we are binding to the acetylcholine receptor and inhibiting the signal from reaching its target. Okay. Well, muscular systems are also targeted by a variety of different types of toxins. So that neuromuscular junction is one place that toxins have evolved to incapacitate. The structure itself is affected in many cases by myotoxins that are based on a phospholipase scaffold, but there are also some very specific ones, and Dr. Hayashi may want to discuss this a little bit further, but myotoxin A and crotamine are these peptide toxins that are very, very potent inhibitors of calcium pumps and perhaps a couple of other target sites in muscle cells, and specifically in the SR, and result in tetanic paralysis, very, very rapid. Well, snakes are not satisfied to target just neuromuscular junctions and muscle function, but in fact, the circulatory system, not surprisingly, is also a site where we can have structural damage occurring to the vessels themselves. We can have cells such as the red cells subjected to damage leading to hemolysis. We can have liberation of particular kinds of blood pressure regulators such as bradykinin, and many of these kallikrein-like serine proteinases are actually liberating bradykinin that results in vasodilation. There are compounds specifically interacting with platelets, interfering with platelet function in a number of different ways. These are primarily disintegrins and C-type lectin compounds. And then there are thrombin-like enzymes that are very common in viper venoms that, in fact, will hit fibrinogen directly and affect clotting via the degradation of fibrinogen. Finally, then there are some specialized, primarily metalloproteinases, but some serine proteinases that can lead to activation of factor 10, and these produce particularly nasty symptoms in humans when envenomated. So a number of different targets or different regulatory sites in animals are in fact targeted by these different venom proteins. And this is just 3 common areas that are commonly targeted. So what I'd like to do now is take you on a historic journey through some of the things that we've worked with and look at venoms, which we can consider bioweapons, and how snakes have in fact evolved mechanisms for production and storage safely. Because if you think about it, this gland here containing all these different toxins we just talked about is not very far from the brain, and there's a potential then for this toxic arsenal to be problematic for the holder of itself. Obviously the snakes exist, they've diversified, so we just need to figure out how they're in fact doing this. So we've just seen there are a variety of different enzymes in particular, as well as toxins that are contained here. And in snakes, these can be stored for a very long period of time. And so snakes at Butantan and in my lab, some of these we've had for many years. Some of the snakes don't get extracted, and so that venom can stay in that gland for many years in some cases and is ready to go at a moment's notice. In addition, these snake venom metalloproteinase compounds are in fact autolytic. If you try to purify these under the wrong conditions, the molecules will chew themselves up. So there are problems with storing this venom in this gland that's on the side of the head. So how do they do that? Is venom activated? Is it stored inactive? Is it stored in a safe fashion? How is that maintained in the gland? And are there specific cellular mechanisms that exist among these specialized glands that allow these snakes to do all this, have the material ready, used in predation, perhaps in defense as well, but not in fact have toxic effects against the holder of that gland, the rattlesnake in this case. So if we look at the venom gland itself, and this was stuff from way back in the day, the venom gland itself is divided into a couple of different regions here, and there's a lot of stored venom here. And so they can hold that venom in the gland for a very long period of time. Most of the cells are secretory cells. If you extract them, there is a very rapid response. They increase in height by about 100% within a couple of days, and they become protein synthesizing factories. Citrate has been noted as a component of venoms for a long time. And we did some work with it a while back and showed that possibly it could be important, but in fact, in vitro, it doesn't at optimal pH for the enzyme, but it doesn't actually inhibit a significant amount of metalloproteinase activity. So citrate doesn't seem to be the most important component here, but we're going to come back to that in a moment. It turns out there are also endogenous inhibitors, and some of these are little tripeptide molecules that are found in the venom, oftentimes at millimolar concentrations. So they're in the venom at high concentrations, but they're relatively low affinity inhibitors. So if we look at some of the metal chelators, what we see is that they're very effective at shutting down metalloproteinases. And these are pictures from, or figures from purified metalloproteinases from a couple of rattlesnakes. If we compare the inhibition by these tripeptides, what we see is it's much lower affinity for these metalloproteinases. So we get out to very high concentrations, we can get complete suppression. But what it does also do is stabilize the venom. And so if we take the purified metalloproteinase, incubate it for a long period of time, what we see is we lose activity. It eventually drops to almost zero. But if it's incubated in the presence of this little inhibitor, it in fact stabilizes and retains activity even under incubation at 37 for up to 4 days or so. So very important as an inhibitor. So, and a stabilizer, I should say. So the other question is, are there cell-based inhibitory mechanisms that occur? And what we see is that there's an unusual little cell called a mitochondria-rich cell. And it occurs in the venom gland at fairly low copy numbers. So there's only about 2% of the total population that are these mitochondria-rich cells. It also cycles with the secretory epithelium as the cells undergo the secretory phase. And so they're cuboidal at rest, columnar when they're secreting or they're producing, and then cuboidal again after they've secreted into the gland. And these cells follow that same pattern of increase, decrease, increase, decrease. So we were interested in looking at those in a little more detail. And it turns out they have some interesting aspects of their morphology. They sit in this little pocket. And in the previous slide we saw, you can see a little top of the cell here, the apex, sticking out like a little button. But there's a channel down here that is continuous with the outside, that are these regions of canaliculi that you can see at the arrowheads here and right along here and this space here. And when I first saw these years ago, I thought this was damaged because I did a bad job preserving the tissue, but it turns out those are actual. Well, it turns out those are very, very similar in their overall morphology to cells that are found in a rather different sort of place. And those are the parietal cells of the gastric pit of the stomach of either mammals, like ourselves, or rattlesnakes. And what we see is this morphology where it's a recessed cell and it has lots and lots of mitochondria, very different from the mucus secreting cells that are in the vicinity in the stomach, or the serous secretory cells that are typical of rattlesnakes. And so that led us to think that maybe we've got something unusual going on here with these things. Maybe they are specialized. And so histologically, we did some work staining these tissues. And what we see is that these cells here, seen here with H&E, and then here with nitro-BT, that is specific for these acid-secreting cells, shows a very localization of these types of activities. Okay, so that was how far we got a number of years ago. And it turns out that if we look at the venom pH, what we see is that the venom is actually produced at and stored at a moderately acidic pH, right about 5.4. And when I've talked with colleagues around the world, it turns out that other vipers, true vipers as well as pit vipers, are right about that same level. Well, if we look at that compared to enzyme activities, and these are a bunch of purified enzymes from the same rattlesnake venoms, what we see is that at this pH, there's almost no enzymatic activity. Well, fast forward then into recent times, and our collaboration with Todd Casto's group in Texas has led us to be able to look at the same system now in a lot more detail. And what we see here in this diagram is that when we have venom or venom glands that are going from rest to 1 day post-extraction, not surprising, we see gene expression of all these different toxin families, and there are 3 listed out here and then combined other minimal ones here. But we see a tremendous increase in gene expression coding for those particular toxins. Not too surprising here, okay? If we compare that to various other types of tissues, though, we see a very different sort of pattern, okay? And so in this heat map, what we see is if we look at all of these different transporters here, so now we're narrowing our view just onto what's going on with particular gene expression patterns. What we see is that in the venom glands, we see very high expression in a number of different types of tissues, or sorry, a number of different types of transporters. And these here are the ones that are comparison of unextracted versus 1 day post-extraction. And so what we see in this region here is very hot colors indicating very high levels of expression. Well, we wanted to look at that in a little bit more detail, and so we actually looked at some of the specific transporters and asked the question, are these being expressed at high levels in these various tissues? And so we first looked at gastric membranes from rattlesnakes, from rats, and from the venom gland, just total membranes here, and asked the question, do we see HK ATPases being overexpressed? And basically what we see is that they're not expressed. So that doesn't look like it's the case. This particular transporter here, and this is involved in acidification of particular tissues, this is not involved in the whole process. If we look at a different type of transporter, and this is a vacuolar ATPase, what we see is that just like in the gastric membrane, in the venom glands, we see that it is very highly expressed. And so looking at these Western blots provides us with evidence that we have a particular type of transporter that is involved in acidifying the venom, could be involved. So this looks like a pretty good one to follow up on. So we did some fluorescence microscopy on this tissue as well. And if you remember that little mitochondria-rich cell that was recessed in kind of triangular shape, what we see is here and here, we see it lighting up specifically with these antibodies designed to recognize those vacuolar ATPases. And in all of those cell types only, and not in the surrounding epithelium, we see those cells lighting up very brightly. Okay, so what we have then is a picture where these mitochondria-rich cells that were enigma for many, many years to functional morphologists, we suggest that they are actually acting as an acidifying cell, that is producing acid to drop the pH in the storage conditions of the gland to about 5.4. Citrate, that has a marginal inhibiting effect, we don't think is overly important as an inhibitor directly, but it turns out that one of the pKa's of citrate is right at about 5.4. And so that means it's going to be a very good buffer for maintaining that pH. So we believe that these vacuolar ATPases are in fact responsible for acidifying it, and the effect is to store the enzymes that potentially could chew up other components and maybe damage the gland and the snake itself, are stored at this inactive state. Further, we have spontaneous activation when we inject it into prey, because now we're going from a milieu that's 5.4 to a pH 2 log steps up. And so, in fact, most of the enzymes are very active at 7.4. Well, like any biological system that's potentially hazardous, there are redundant mechanisms for maintaining this. And so what we believe is happening is that this is a proximal mechanism that allows for the venoms to be expressed and stored for many years in a stable environment. The peptide inhibitors, the tripeptides, also contribute to this whole thing. And then there has been some work done a long time ago showing that some of the metalloproteinases are expressed initially as zymogens, but they seem to be clipped shortly after secretion from their cells. So this is probably not a major mechanism for safe storage. The thing to me that was very interesting is that morphologically and functionally, the cell type that's found in the venom gland is very similar to what we find in the gastric pit in the stomach of many different types of vertebrate animals. And to me, that just indicates another link here between the trophic roles of venom, how venoms are used for feeding, and digestion. And so it's kind of an interesting little aside there that these roles seem to be, again, conservatively using the same kind of mechanisms for doing particular things. Okay. A difference here, though, is that instead of using an HK transporter, which we see commonly in the gastric pit, we have instead this vacuolar ATPase as a major transporter resulting in acidification, again, limited to those mitochondria-rich cells. Okay. So, well, what I'd like to do now is take a look at kind of ecological roles of venoms and also talk about how that has, in fact, been important towards the evolution of a particular family of toxins, and that is the three-finger toxins. So we know that snakes use these venoms to incapacitate prey. So here is a Sonoran coral snake eating a little ground snake here. And here is a rattlesnake. You may not have seen it. It's rather cryptic. And right there is an animal that's dispatched, a little thing called a chipmunk. And in fact, it shortly thereafter consumed that. So the venoms we know are important for doing this, stopping prey, immobilizing prey, allowing the snake to handle it. Okay. But the venoms are not the same across the board. We've seen that in the intro type of materials that we talked about earlier. So what we get are variations of genes occurring via, we believe, first gene duplication. These are overexpressed in the venom gland, and a variety of people, including our collaborators, have shown this over and over. We can get subfunctionalization, but more importantly, neofunctionalization of some of these duplicated genes, and this then can lead to expansion of gene families, giving us a wide variety of different options that we see in the venoms of various snakes. So if we look at particular species, we may see as many as 10 to 20 or more transcripts for three-finger toxins or serine proteinases or things like that. So we know that some of these gene families are extremely abundant. That is, it's expanded tremendously. Okay. Well, we talked about this already, that there are a number of targeted systems, and one of these are the neuromuscular junction. In particular, I want to look at this one here, the acetylcholine receptor, which is postsynaptic, because this is where a number of different three-finger toxin subfamily groups have in fact targeted for knocking them out. Okay. So the acetylcholine receptor in one form or another is found in all animals. Major importance for neuromuscular communication and overall muscular function. And toxins typically then will bind to the alpha subunits and incapacitate this structure, and the receptor is no longer functional. The result is that we get flaccid paralysis of the prey because the neurons are saying contract, contract, but that signal is in fact blocked. Okay. Well, let's look at those three-finger toxins. They're kind of smaller molecules, so as far as diversity of toxins, they're relatively small. They're tightly tied together by intramolecular disulfides, shown in gold in this diagram here. And there are 4 or 5 of these typically. The 5th one is down here in loop 1. And that gives them that characteristic canonical structure, the 3 fingers that are tied together at the head group right here. Okay. We find these pretty broadly distributed across various species of snakes. The typical activity that most people have focused in is as curare mimetic toxins, alpha neurotoxic activities, binding to that acetylcholine receptor, as we've mentioned, that is skeletal muscle, and leading to this flaccid paralysis. But it turns out this is one of those scaffolds that has been very, very useful for a variety of different types of pharmacologies. And so if we look across the board, we see a whole bunch of different things. And below this line here, if you happen to be a salamander, this type of molecule is used as a receptivity pheromone and also as a modulator of limb regeneration. Salamanders can regenerate limbs. So this is a very, very useful molecular scaffold, and various life forms have in fact played with it to do a wide variety of different things. Toxic activities, or very important activities here involved with modulating structures and reproductive activities. Okay. These have also, in fact, resulted from gene duplication. There have been a variety of different ways, attempts to try to explain what's going on. It almost seems like there are like cassette-like movements of material, of genetic material, this so-called asset or segment switching in exons. And point mutations then, after we have gross level changes, can fine-tune these kind of interactions. Okay. What we see is that the structurally important residues tend to be highly conserved, maintaining that 3-finger motif and 3-finger morphology. But surface residues that are involved with ligand recognition and binding are in fact basically open to variation. If you happen to be involved in snakebite treatment, these are most problematic because these things bind really, really tightly, often in the sub-nanomolar range. And so they basically knock out these receptors, and the receptors have to be turned over. They're small, so they diffuse, or they're transported very, very rapidly through systems. They're often found at very high concentrations. And in cobras, for example, we find that the venom in these 2 common species, Indian cobra and monocle cobra, may account for 15 to 20% of the total venom proteome. And this is a single toxin, not a toxin family, but a single toxin, cobra toxin. Untreated, of course, this often leads to fatal conditions, often by respiratory paralysis. So there's been a lot of interest in these particular toxins because they're problematic in many parts of the world where elapids are very, very common. So we see these things in a variety of different snakes. And so here is a monocle cobra. Here is a sea snake, one of the Laticata species. And here is a rear fang snake. And what I'd like to do is focus in on some of the toxins from these guys. Okay. So these are 3-finger toxins. We have 3 overlain here, superimposed. Cobra toxin from cobras, Fulgema toxin and Denmo toxins, both from different rear fang snakes. And what we see is that their overall fold is very, very similar. If we look at their toxicity to mammals, sure, cobra toxin is very, very toxic. But if we look at a number of these from different species of rear-fanged snakes, we see that they're essentially non-toxic. And most pharmacologists have stopped here. These are not interesting because they don't have much effect. We were more interested in them from a biological perspective and how the snake is using these for the prey. And so we looked at their effect on other animals, non-model animals. They're essentially non-toxic to mice, but in lizards, in this case a gecko, this toxin is exceptionally toxic. And in small birds, they're very, very toxic and approximating the toxicity of what we see in cobra toxin. So here we have an enigmatic situation, non-toxic in mammals, highly toxic in lizards and birds. And we've seen this repeated in both dimeric and monomeric toxins from a number of different species. Most recently, we've worked with one that also has a variant here, and it's got some structural differences from the other ones, and it in fact shows the opposite pattern. That is, it's non-toxic to lizards, but it's in fact moderately toxic to mammals. And these are found in the same venom, the Amazon puffing snake, Spilotes sulphureus. Okay, so we're interested in trying to figure out how that actually occurs. And I'm getting a little bit short on time here, so I'm going to move through these a little bit more rapidly. Structurally, they're very, very similar, but in fact, what we see is that if you look at the overall sequence, they don't share much of that, for 40 or 50% identity. And there are particular areas that we've identified in these taxon-specific toxins that affect lizards, but not mammals. And in fact, they are highly conserved in all of these different toxins. So, in the one that shows that reversed situation, what we see is that we have a substitution in one of those invariant motif regions, and in the tip, we also see a deletion. And so there's some structural differences here that lead to overall differences, as you've seen colored here. These are all highly toxic to birds and lizards, but are non-toxic to mammals. This one is in fact toxic to mammals, but non-toxic to lizards.
Stephen, I'm sorry, may I ask you a question?
Yes.
When you say toxic, what do you mean about toxic? It's toxic lethal, or is toxin for some specific effect? Which kind of toxicity?
Lethal toxicity is what we're looking at here. Okay.
Is that for rodents?
Sorry?
It's only for rodents? Which kind of animal?
Well, the ones that we have from colubrids primarily are non-toxic to mammals, but very toxic to lizards and birds.
Okay, I was going to ask if there is a big difference between birds and rodents.
Well, yeah, it's an interesting feature that we haven't completely worked out yet, as far as what it is that is producing this toxicity. What we see, though, is a very, very high level of conservation of the overall scaffold, but diversification of function towards toxicity towards particular prey types. Okay? And it turns out that if you look at the animals that the snakes are feeding on, these tend to be the highly preferred prey items. So they're feeding on birds and lizards, whereas things like rattlesnakes and cobras may be also feeding on mammals. Okay? This one in particular is an interesting one because it has a very stripped down venom, and the proteome consists of only, well, majority of the proteome is three-fingered toxin rich. It's got a couple of different types that are taxon specific towards either mammals or towards lizards. It's not a constricting snake, and so it's using its venom to incapacitate these two different types of prey. So hopefully over the last 45, 50 minutes, I've convinced you that venoms and venom glands are an interesting model system to look at a whole bunch of different types of biological phenomena, including gene regulation, secretion, and storage of these bioweapons, these molecules that are very toxic. They're a rich source of materials for studying structure and function, and with the three-finger toxins in particular, that is an important component. I would argue that understanding how three-finger toxins have evolved and diversified, in order to do that, we're going to have to understand what's going on with these rear-fang snakes, and only a handful of those have actually been looked at in any detail. Dimeric structure, I have a student that's finishing up his work on this, and he's starting to get at answering how important the dimer is composed to, as opposed to a monomeric structure. The nice thing is that if you're interested in this, there are over 500 species of rear-fang snakes that still haven't been explored at all globally. So regardless of where you are around the world, there's an opportunity to make important contributions. So I'd like to acknowledge support from a number of different funding agencies that have provided us support over the years. And of course, collaborators of a variety of types, Cassie Modal in particular, doing a lot of the molecular work in conjunction with Seth Fritz and Mernalini, very important students and collaborators here. Todd Casto, pictured here in the field, and his graduate student Blair Perry, absolute tremendous genomics wizards. I've had a number of students over the years that have contributed to various parts of these studies as well. And my current group is here with a couple of people that weren't there for the picture. And so all of their help has been, of course, tremendously important for that. Just a little advertisement. And Dr. Hayashi has contributed a chapter to this book that is due out very, very soon. And with that, I'd like to thank you for your attention. And if we have time, I'd be happy to try to answer any questions that you might have. So thank you for your attention.
Thank you very much for your presentation, Stephen. It's very exciting and very nice pictures.
Well, thank you.
So I do have a question for the audience. And
Yeah.
Asking if it is possible that the venom is used for pre-digest the prey. To some extent, it's helping for the digestion of the prey, and not only for killing or, paralyzing the prey.
Well, that's a very good question, because that was basically the whole focus of my master's thesis. And at that time, the hypothesis that I was working with was that it is very important to pre-digesting the prey. Since that time, there have been a number of studies looking at the level of digestion of envenomated versus non-envenomated prey. And the suggestion from those studies, and there's been several, is that the presence of those digestive enzymes is not facilitating digestive efficiency. Now, there are some limitations to the way that those studies were constructed with how you interpret that. What we do know is that if an animal is bitten by a rattlesnake or another species that has high metalloproteinase activity, there's a lot of structural degradation. In addition to the death of the animal that eventually results, tissue is broken down. So it's hard not to believe that the metalloproteinases and some of the other components are contributing to pre-digesting or tenderizing or conditioning the prey to make it easier to digest. Okay, but that may not be reflected in these metabolic rate studies that were in fact conducted that suggest that venom doesn't have an effect. So I think it does probably have a very important effect, but the jury is still out.
Yeah, you are focusing on metalloproteases, right? I do have my own opinion. Actually, while I was in Butantan, I was working with those bradykinin potentiating peptides, the BPPs.
Yeah.
And those BPPs, actually, they are not produced only in the venom. So I don't know if you have read some of my publications, but I showed that the same BPP precursor protein can be expressed in other tissues, physiological tissues of the snake. So for the Brazilian pit viper, Jararaca, I have seen that the same RNA is expressed or is present in the brain and also in pancreas. And for a while, I was believing that maybe those BPPs in pancreas was somehow signalizing or being signalized by the venom after the digestion. So I think that there might have some endogenous or physiological function for all those toxins, and they are not toxic only because they are underexpressed. So in the gland venom, they are just overexpressed, and then when you have a high amount of anything, it can be venom. Yeah,
no, that's one of the truisms that we see in venom compositional analysis is that there are analogs or paralogs or homologs found in a wide variety of different types of tissues. And a number of the publications that we've had with Todd Casso's group have shown the expression across the board of these things, but particular subclasses of metalloproteinases, of probably some of the BPPs as well, and certainly things like myotoxins and other compounds, both the PLA2s and the peptides, are way overexpressed in the venom gland relative to their expression levels in other tissues. So, but yeah, it is a common feature that we see across the board, that these toxins are not newly evolved toxins out of nowhere, but instead they're co-opted regulatory compounds of a wide variety of different types. And that includes even the three-finger toxins. And so, you know, those were probably before the evolution of venoms, which was probably a very, very long time ago. I mean, hundreds of millions of years ago. These were physiological compounds, housekeeping compounds. And so, yeah, so it's not surprising we see them in various tissues.
Yeah, so actually my point of view is that even those three peptides that you claim that could be inhibitor or stabilizer of pyroglu, asparagine, and tryptophan, and they are also actually usually the N-terminal of those BPPs. And I know that they are freely present in the venom. So my point is that actually if they do have those same peptides in physiological system, they might have a receptor. So maybe those peptides or small peptides present in the venom, once they get into the stomach of this snake, they are going to signalize something else, acting on a specific receptor. So this can trigger some release of digestion liquid or whatever, or internal enzymes, and not exactly about the MPPs present in the venom.
So are you thinking that they would be transported out of the prey into the snake tissues?
Yeah, so actually in snake tissue, they are going to have those receptors because they are already expressing those peptides. Yeah. So they have activation of some internal signaling in the snake that can, you know, tell the snake that they are eating something. Yeah.
Well, that certainly could be the case. If the venom, though, is internal, it's going to take a while for it to break down through the tissues and the stomach acids and digestive enzymes to break through the tissues and allow that to get into the tissues of the snakes. So there would probably be a delay of maybe even 24 to 48 hours before that happened. But yeah, that could be, that then is important as signaling upregulation of some digestive process or something like that.
Okay, I'm sorry, I get so excited with your presentation and I do have so many questions, but I have to go back to this. So there was a student asking why some species of animals are not affected by the venom of other snakes.
Well, that's a very good question too, and I've got a student right now that's looking at resistance to venoms in mammals and in other snakes, and a number of people in Brazil have actually addressed those same kind of questions, and some of them are actually contributors to the book as well. In some respects, we can think of there's an evolutionary arms race or coevolution between predator and prey, and because we're dealing with things that are operating physiologically, biochemically, we can have a physiological response that can counter the effects of those toxins. And we have, being vertebrate animals, whether you're humans or mice, there are a number of endogenous inhibitory compounds. Alpha macroglobulin is one of them, that are basically taking care of what I call rogue enzymes. So, for example, the way that the blood clots is highly regulated by a whole series of cascading steps. And you want those to be very active locally, but shut down throughout the system so that we're not throwing clots all through them. And so part of that is regulated by endogenous compounds like that are in the blood. And so what seems to be happening is some animals have the capacity to overexpress some of these compounds. They may also be subject to selection favoring modification of their binding properties so that they recognize particular metalloproteinases or serine proteinases or maybe even three-finger toxins and bind them and provide some immunity to the animal. So in the case of the, go ahead.
No, you can finish, sorry.
And I was going to mention, in the case of three-finger toxins, these are binding specifically to receptors that are found in one whole category of prey, lizards and birds, versus a very different type of vertebrate animal, the mammals. And precisely how that's occurring, we have hypotheses, but we're not really sure. So.
Okay, I'm sorry for interrupting you. I thought
No,
you have.
that's fine.
Okay, the other question is from Livia Maria, but I'm not really sure if you will understand the question. So if it's not clear, we can just ask her to ask again. But the question is, what kind of bioinformatics tools are used to study the potential application of enzyme and other compounds in the pharmaceutical area? So how can you recognize those that you can use in pharmaceutical?
Well, I guess the big disclaimer is that my bioinformatics background is very minimal. And so I rely on the expertise of my collaborators. Cassie Modal was a PhD student working with me, then went to Dr. Kinney's lab in Singapore, and she became a very good bioinformaticist. And so those kind of questions I largely left on her desk. So as far as applications of some of these toxins to potential pharmaceuticals, we've done a little bit of molecular modeling and such, in silico, but not a real extensive amount of that. Again, I have a PhD student that is hoping to finish up soon, and he's interested in doing some of that work. So.
Yeah, okay. I hope Livia is happy with this answer. If not, please write the question again. So I just have the question is from Juan David, and he says, thanks for your awesome presentation. And he would like to ask you if you think that new previously unidentified protein scaffold, so still unknown protein scaffold may still be hidden in venom from here fanged snakes. I don't know why this is specific snake, but anyway.
Yeah. So are there some novel scaffolds out there that we haven't yet discovered?
Yeah, and they could be of some useful for medical bias that exist towards vipers and elapids. So I think that his concern is about some specific snakes, because there is a lot of elapids and vipers, but not so many about other snake species.
Yeah. Well, I think that the colubrid snakes globally, the rear fang snakes, are still a very productive area for looking for new compounds and interesting compounds. But one of the things that is apparent is that snakes are fairly conservative with the selection of things that they've really grabbed onto and diversified. Okay, so the types of compounds that we see as the most predominant in any particular venom, you see the same basic players again and again and again. Now, there are lots and lots of compounds in the venoms that are expressed in very, very small amounts. And from a perspective of what is the biological relevance of some of those compounds, they're probably not important to producing the symptoms of envenomation. It's possible some of them are biologically, super active, highly active, but it's unlikely that they're contributing as much if they're only 0.01% of the total proteome as something that's 20%, for example. Just to give you an example of the differences, in our part of Colorado, the prairie rattlesnakes venoms from adult snakes from a den site that we've worked at, the myotoxin A component of those venoms can comprise, up to 35% of the total venom protein, just that one particular molecule. And so they've really heavily invested in that direction, if you will, evolutionarily. So so I think we're going to see a lot of the same kind of molecules, same kind of scaffolds as the abundant proteins. Now, if you're interested in mining the less abundant proteins and really trying to identify those, that's going to require some kind of cloning and overexpression system in order to work with the material pharmacologically. But that could be potentially very, very interesting and illustrative. And as far as novel compound leads, that could be more productive than going in and asking what are the dominant components of this venom, okay? Because it seems like we're seeing the same kind of things showing up in snakes, whether they're rear fang snakes from Africa, from South America, from North America, or elapids and vipers from other places around the globe. We're getting differences in the overall abundances of a variety of different proteins, but we're seeing the same kind of players as the major players in the venom again and again. Okay.
Yeah. So if you allow me, in my point of view, those with a very low concentration in the venom can be identified if they have very strong pharmacological effect. So if there is a specific biological effect that you can trace, then it makes it easy to identify them. And then you can express by recombinant or using synthetic biology. So there are different ways to obtain. And now, speaking about the composition of the venom by itself, some of the toxins like crotamine, it can represent up to 25% of the dry weight of the venom. But even though it's not really important for the envenomation process, so why it's so evident in the venom? Do you have any idea?
I'm sorry, you said it's 25% of the venom, but it's not important for envenomation? Yeah,
it's not that important. You will never see any rodent or a rat or mouse with the paralyzed because of the crotamine. You can just see it after purification, not using the crude venom. So it does not have to be important for the envenomation, but it's important for the accident, the human accident. So if you use a vaccine against a venom without crotamine, then you cannot protect it for biting with crotamine. But for the envenomation by itself, for the prey, I have never seen any specific effect, biological effect.
This is native species of prey?
I'm talking about rats and mice.
Inbred strains or wild animals?
Actually working with experimental animals in the lab, not in wild animals. But even for wild animals, if you just observe the nature, they are not dying of the paralysis. They are usually dying because of phospholipases. It's a neurotoxic effect. Yeah.
Well, that venom obviously has that presynaptic neurotoxin too. I know that in our prairie rattlesnakes, when they bite a lab mouse, that lab mouse is paralyzed almost instantaneously. And so for them, that's very, very important. I'm surprised that crotamine in lab mice doesn't have that same effect because the sequence is almost identical.
Yeah, I'm talking about crude crotamine. Purified crotamine has effect in lab animals with different sensitivity depending on the strain. I'm talking about venom. If you use the crude venom, the crotamine effect does not make any difference. You don't see crotamine effect using crude venom.
So you don't get hyperextension and immediate paralysis?
No, it dies before due to the neurotoxic effect of phospholipase.
Yeah. That's curious. Yeah. Like I say, with the crude venom, we don't see that.
Yeah. So. Anyway, I will go back again for the audience because there is so many questions. One of them is from Maria da Graça Salomão from the Tantan Institute, and she's just saying excellent presentation. Congratulations. There is one from Juan Gutierrez. And do you think venom could display some degree of phenotypic plasticity on response to environment pressure? For example, prey availability.
So, for example, if you have a single snake and it feeds on this type of prey or that type of prey?
I suppose, yes, if Juan
Yeah.
Gaultier could improve this question, we will be happy.
Yeah.
Really sure.
Well, there's been a lot of discussion about whether or not you can have this kind of epigenetic effect modifying the venom composition. Now, we know that venoms do change in composition for many species when they're newborn to adult, and there are compositional changes that can be pretty profound. But from the limited amount of work that's been published, and from a little bit that we've done that hasn't been published, if you feed lizards to one group and mice to another group, you don't see a very profound effect. There's one paper that was published that showed in one snake that there was a tendency towards overexpression of phospholipase, I think, in the mammal-fed group. But other than that, the effect on the individual snake doesn't seem to be particularly profound, regardless of the type of food. So that epigenetic effect or the regulatory effect on it directly, there doesn't seem to be a direct feedback there.
But there's another way to looking at this, right? Like here in Brazil, we have the pit viper, Bothropis jararaca, that's generation of those BPPs. This in continental area, they have a different composition of the venom compared with the bothrops that live in islands, like Bothrops insularis, that only eat birds. Therefore, somehow, what they are eating are influencing the composition of the venom. Don't you believe in that? It's like
I agree.
Like the Crotalus here in Brazil, the red snake here, has a specific composition that's closer to Bothrops in North America than Crotalus in North America. The composition are completely different, so they are eating different stuff. No?
Yeah. I think there is a direct association between venom composition and prey type. Okay, but I don't think that an individual snake has the capacity to change the expression pattern of its venom if it's fed this birds versus mammals or something like that. Okay, that doesn't seem to be occurring.
Yeah, it's a kind of long-term adaptation.
Yeah.
So it's not seeing just one single individual. But that's true, even in just single individual, if you maintain that in captive, they are going to change the amount of they produce. Depending on the age, sex, and in captive, since the feeding is, it seems that the toxin amount in the venom and also the amount of the venom that is produced is different compared when they are in the nature.
Well,
adaptation.
Yeah, I'm not sure that I agree with that 100%. I think things do change a little bit, but from our experience, at least, snakes that we've had in captivity for a long period of time, for a short period of time, they show almost no difference. For a long period of time, we see very little difference in extractions of a snake that were taken 10 years apart. So to me, that suggests that the effect of captivity and feeding regime that you've imposed on the snake and now can't feed on whatever it would eat in the wild doesn't have a major effect on venom composition, as long as it's nutritionally sufficient, not starving or something like that.
Yeah, it might have some also effect of the snake type, the species. So depending on the species, they have faster, and the other does not care if they are in captive or not. But in addition to this discussion, there is another question from Natalia. So she's asking if the snake venoms present plasticity or could adapt to the prey on an ecological level, and if these features have been seen only in the evolutionary way. Does this adaptation or features of plasticity is only for evolutionary way, or it can present some adaptation or plasticity could adapt to the place? So on ecological level, if it's possible to have the change or adaptation of the snake.
Yeah.
end of the
Yeah.
day. I think it is.
Well, I think one of the basic questions is how fast can venom composition change, right? And what are the factors that affect it? And there have been a number of people, including us, that have tried to look at that question. And it's interesting that a lot of different factors seem to be minimally important. There's some studies that suggest that things like overall environment and climate may in fact be linked to compositional trends and shifts and things like that. But I haven't seen any studies that have really been here. This is the factor that shapes it directly. It's more kind of a much softer kind of a effect. That is, it's not a black and white effect that, you know, if you eat this prey and shift over to this, then the venom composition will change in a particular direction. So I think.
Yeah, go ahead.
No, I was just going to say that I think these kind of things over evolutionary time, we do see these kind of transitions. The problem, of course, is that we're not long-lived enough to track these things over a long period of time, and that's one of the limitations.
Yeah, it seems that this matter really attracts a lot of attention. So there's a couple of more questions, adaptations. So we skip them. And I would like to ask you if you can just go back to your slides and show your new book, so the people can.
Oh yeah.
Making a kind of advertisement for you. Yeah. So please, guys, buy this book. So you're going to find out our answer for your questions. And I'm pretty sure that Dr. Stephen is ready to answer any further questions you have. So you can just send to me, and I can forward for him. And for later, we also share the recording of this seminar, so you can watch it again anytime you want. So on this point, I would like to thank Dr. Stephen for accepting my invitation for this awesome presentation. It was very exciting. A lot of questions. So if you want to give some last words, just take your time. Yeah.
Okay. Well, I'd be happy to answer any questions again if I can. And if you have questions that come to mind later today, tomorrow, feel free to drop me an email. I'd be happy to answer anything like that too. So yeah, no, it's been my pleasure. My only regret is that I'm not there in person, because I would love to be in Sao Paulo and see your university and visit with you face to face. So
actually, you are already invited. You can come anytime. You will be welcome. Okay. We just have to get off of this pandemic situation. That's the great
thing. And I'm concerned that this is going to last a lot longer than any of us want, and any of us are predicting. But hopefully things will get better. You are entering wintertime, or autumn, I guess, and there's a possibility that severity will increase a little bit. You know, we've hit a situation where caseload has dropped a little bit, but starting to spike back up again. So it's hard to predict, but I would love to visit again. As I mentioned, I've only been to Sao Paulo once. I did experience, though, your traffic jams there. Dr. Inácio Unqueira actually took me out to the beach at the Atlantic coast. And on the way back, I think we were stuck in traffic for almost 12 hours to return. So
really,
it reminded me of Southern California.
I'm always in the opposite way. So where I know that it's going to be, I'm going the opposite way. So you go to the mountains in summer, and you go to the beach in winter. That's the best way.
Yeah.
Never do the same as everybody. Otherwise, it's awful.
I try not to, but sometimes you can't help it. So
next time you come, I'm quite good for skipping traffic.
Okay, I'll take you up on that then.
Okay. So thank you again.
All right.
Let's just try.
Again, my pleasure.
Yeah, and we can keep discussing, and maybe you can introduce me, Mark, your friend, Mark Thomas. Was that?
Sure. Yeah, I will try to do that.